In my opinion, CRISPR is one of the most important scientific discoveries of all time. Back in 1987, researchers at Osaka University were studying E. coli bacteria and noticed something really odd in the bacteria's genome. There were 5 identical segments of DNA in there that had short, non-identical sequences between them. And a bunch of other scientists started finding these weird segments of DNA in all sorts of other bacteria. Like, they found it in so many bacteria that they decided it's gotta be important, and it's probably time to give it a name, too. And they settled on the acronym CRISPR, which stands for Clustered, Regularly Interspaced Short Palindromic Repeats. But no one really knew what was going on with it, much less why it was so important. By 2005, there were loads of databases with these genomic sequences. And scientists found out that segments of DNA in between the repeated parts are actually virus DNA. So bacteria DNA has virus DNA inside its own genome, but only in the segments between the short palindromic repeating parts. But then a scientist, Eugene Koonin, was like, oh shit, I think I know what's going on here. He was like, this is kind of like a mugshot of viruses, like a storage facility of threats. Because bacteria are always getting attacked by viruses. It's estimated that viruses kill 20% of bacteria in the ocean every day. A virus will attach itself to a bacteria and then inject its own DNA in there, and then the bacteria becomes a virus-making machine until it fills up and explodes with them. But if it survives, it sends out an enzyme to cut up the virus DNA and then shoves it in between those palindromic repeating parts. And the next time it encounters the same type of virus, it has a mugshot to compare it to. And that mugshot gets held in a specific protein called a CRISPR-associated protein, or Cas protein. And Cas proteins have these really cool molecular scissors that precisely cut up invading DNA. It unzips the virus DNA and compares it to the mugshot. And when it finds an exact copy, the Cas protein locks into place and the molecular scissors cut it up. And that's all pretty cool, but we still haven't gotten to the revolutionary, world-changing discovery that is modern CRISPR research. In 2012, biochemical researcher at UC Berkeley, Jennifer Doudna, and her research partner, Emmanuelle Charpentier, were the first to propose that you could use Cas9 proteins as a useful tool for gene editing. Jennifer Doudna said that when the full impact of that hit her, the hairs on the back of her neck stood up. So instead of using it for defense, we can use it for offense. And she was like, why don't we just replace the mugshot? And instead of attacking viruses, we can get it to attack the genes responsible for things like sickle cell anemia, Huntington's, hemophilia, or cancer. Just put a gene responsible for a disease in the Cas9 protein and it gets cut right out. It also turns out to be really easy to put your own edited gene there into the site where you cut. You just have your good gene hanging out right around the area where the Cas protein is. Because your cells have repair enzymes that scour your DNA for breaks or damage all the time. And usually if there's a break, the repair enzyme will quickly put it back together. It might cause a small mutation, but the DNA is complete again and can continue transcribing. But if you have that good DNA hanging around, the repair enzyme will just shove it into the newly created space and bam, you don't got sickle cell anymore. It's quite a bit more complex than that, but that's the gist of it. CRISPR is universal. They haven't found a single organism that CRISPR don't work on. And the applications are almost limitless. And Jennifer Doudna and Emmanuelle Charpentier won the 2020 Nobel Chemistry Prize for their work.